should fetal stem cells be used in researching cures for diseases such as Alzheimer's? I chose this topic because I found it interesting as a young woman before I have children. The production of blood cells is a lifelong process that is carried out by hematopoietic stem cells, which are generated and maintained in hematopoietic tissues. Hematopoietic stem cells constitute a very small pool of cells that can be functionally defined by their self-renewal properties, their differentiation into all types of mature blood cells, and their ability to reconstitute the marrow of irradiated host. Although hematopoietic stem cells transplantation is a critical component in the therapy for the treatment of hematological and non-hematological pathologies, shortage in hematopoietic stem cells from human leukocyte antigen matched donors continues to limit their use and drives efforts to expand existing sources as well as to identify new sources of hematopoietic stem cells. There are many ethical concerns surrounding stem cell research, and it varies depending on the origin of the stem cells. Human stem cells used in research are termed adult stem cells, embryonic, and germline based on how they are obtained. Stem cells retrieved from umbilical cord blood following birth have similar properties to adult stem cells and are also used in the treatment for some leukemias. Embryonic stem cells are collected from the inner cell mass of an early embryo, thus destroying the embryo. These cells are unspecified, meaning that they have the ability to develop into any type of cell in the body except for the placenta. They also are able to renew themselves indefinitely in the laboratory. Embryonic germline stem cells are immature cells that can become sperm and egg cells. The goal of stem cell research is a relief of human suffering, which is ethically good. Potential benefits of interventions developed through stem cell research include therapies for regenerating various tissues that are diseased and using embryonic stem cells to develop an entire organ to be used for transplant. The main controversy surrounding stem cell research focuses on the use of embryonic stem cells because the process of obtaining these cells results in destruction of the embryo. There are two main objections to stem cell research. The first focuses on whether the destruction of the human embryo constitutes the killing of a human being. The second objection raises the concern that if one considers that research on treating human embryos is not wrong in itself, would it lead to a slippery slope of treating human life as a commodity through practices that are dehumanizing, such as using fetuses to spare parts? Views about human life begins whether the early embryo is considered a person with moral status or at the heart of the ethical deliberations related to embryonic stem cell research. Those who believe that human life and personhood begins at the conception hold that destruction of a human embryo constitutes killing. Thus, stem cell research using embryonic stem cells is never morally allowed. Those who hold the view that the embryo is afforded personhood at the point of viability may consider the use of early embryonic tissue permissible for research. Those who suggest that sentience, life history, and memory are requirements for personhood argue that the early embryo does not possess personhood. Thus, it is morally acceptable to do embryonic stem cell research using the early embryonic tissue if this may potentially benefit human health. Other ethical concerns related to stem cell research include issues of obtaining informed consent from the donors, the potential of combining embryonic stem cells and cloning technique, the potential development of embryo farms, and the issues of ownership of the biologic materials derived from the cells. Because outcomes of stem cell research have the potential to alter human genetics and the human life cycle, there can be social and environmental consequences of extending human life, as well as a shift in our understanding of what it means to be a human. The understanding of Alzheimer's disease pathogenesis is currently limited by difficulties in obtaining live neurons from patients and the inability to model the sporadic form of the disease. It may be possible to overcome these challenges by reprogramming primary cells from patient-induced pluripotent stem cells. Alzheimer's disease is a progressive neurodegenerative disorder which impairs the memory and intellectual abilities of the affected individuals. Loss of episodic as well as somatic memory is an early and principal feature. The basal four-membrane cholinergic system is the population of neurons most affected by the neurodegenerative disease. Extracellular as well as intracellular deposition and beta amyloid or a beta protein, intracellular formation and neurofibrillary tangles, and neuronal loss are the neuropathological hallmarks of Alzheimer's disease. In the last few years, hopes were raised that cell replacement therapy would provide cure by compensating the lost neuronal systems. Stem cells obtained from embryonic as well as adult tissue and grafted into the intact membrane of mice or rats were mostly followed by their incorporation into the host parenchyma and differentiation into the functional neural lineages. In the lesion brain, stem cells exhibited target migration towards the damaged regions of the brain, where they were engrafted, proliferated, and matured into functional neurons. Neuroprecursor cells can be intravenously administered and yet migrate into the brain, damaged areas, and induce functional recovery. Observations in animal models of Alzheimer's disease have provided evidence that transplanted stem cells or neuroprecursor cells survive, migrate, and differentiate in cholinergic neurons, astrocytes, and oligodendrocytes with amelioration of the learning memory deficits. Besides replacement of lost or damaged cells, stem cells stimulate endogenous neuroprecursors, enhance structural neuroplasticity, and downregulate pro-inflammatory cytokines and neuronal apoptic death. Stem cells can also be genetically modified to express growth factors in the human brain. In the last years, evidence indicated that the adult brain of mammals preserves the capacity to generate new neurons from neural stem or progenitor cells. Inefficient adult neurogenesis may contribute to the pathogenesis of Alzheimer's disease and the other neurodegenerative disorders. An attempt at immobilizing this endogenous pool of resident stem cells provides another attractive approach for the treatment of Alzheimer's disease. Studies in patients with Alzheimer's disease indicated decreased hippocampal volume derived from neurodegeneration. Intriguingly, many drugs, including antidepressants, lithium, acetylcholinesterase inhibitors, and ginkgo biloba, were able to enhance impaired neurogenesis disease. This paved the path towards exploring the possible pharmacological manipulation of neurogenesis, which would offer an alternative approach to the treatment of Alzheimer's disease. No stem cell treatments for Alzheimer's disease are yet available. Many different types of neurons in all parts of the brain are affected by the disease. This poses a complex problem if we want to replace the damaged brain cells. For example, one approach might be to transplant neuronal stem cells into the brain of an Alzheimer's patient and hope that they would make new, healthy neurons. But even if healthy, working neural stem cells were available and could be transplanted safely, they would have to achieve several difficult tasks before any therapeutic benefits may be seen. They have to travel into multiple areas of the brain where the damage has occurred, produce the many different types of neurons needed to replace the damaged or lost cells, and do this in a way that enables the new neurons to integrate effectively into the brain, making connections to replace the lost part of a complex network. Despite these significant challenges, scientists have been actively engaged in research on stem cell transplants in mice, and studies have shown some benefits. This is very early stage research, and there are still many questions to be answered. For example, many scientists are concerned that the brain may lack the ability to integrate new neurons properly once Alzheimer's has taken hold. Another concern is that transplanted stem cells might be damaged by the amyloid proteins that build up in the brain, meaning a transplant could only have a temporary effect. Much more work is needed before the findings could be applied to developing a therapy for human patients. Another possible approach to stem cell therapies might be the use of certain types of stem cells to deliver proteins called neurotrophins to the brain. In a healthy brain, neurotrophins support the growth and survival of neurons, but in Alzheimer's patients, neurotrophins production is low. Neurostem cells produce neurotrophins and so might offer a route to solving this problem. To test this theory, Scientists bred mice with the key symptoms and characteristics of Alzheimer's, such as memory impairment. They then injected neural stem cells into the brains of the mice and observed some improvement in memory. Further studies are now taking place to understand this effect, but the approach has not been yet tested in human patients. Many scientists believe that Alzheimer's patients will benefit from stem cells in a different way before the development of potential cell transplantation therapies. By using stem cells derived from Alzheimer's patients to grow large numbers of brain cells in the lab, scientists can study the disease and search for new drugs. There are many pros and cons to stem cell therapy. Pros include that it offers a lot of medical benefits in the therapeutic sectors of regenerative medicine and cloning. It shows great potential in the treatment of a number of conditions like Parkinson's disease, spinal cord injuries, Alzheimer's disease, schizophrenia, cancer, diabetes, and many others. It helps the researchers know more about the growth of human cells and their development. In the future, the stem cell research can allow the scientists to test a number of potential medicines and drugs without carrying out any tests on animals or humans. The drug can be tested on a population of cells directly. The stem cell therapy also allows researchers to study developmental stages that cannot be known directly through the human embryo and can be used in the treatment of a number of birth defects, infertility problems, and also a pregnancy loss. A higher understanding will allow the treatment of abnormal development in the human body. The stem cell therapy puts into use the cells of patients' own body, and hence the risk of rejection can be reduced because the cell belongs to the same human body. The cons of stem cell therapy include the following. The use of stem cells for research involves the destruction of blastocytes that are formed in the laboratory fertilization of the human egg. The long-term side effects of the therapy are still unknown. The disadvantage of adult stem cells is that cells of particular origin and generate cells only of that type, like the brain cells would generate only brain cells, and so on. If the cells used in the therapy are embryonic, then the disadvantage is that the cells will not form the same human body and there are chances of rejection. The stem cell therapy is still under the process of research, and there are a number of things that need to be established before it is used as a treatment line.